Welcome and a very good morning, afternoon, and evening to all of you from the different time zones from which you are participating. My name is Johannes Burgers. I teach English at Ashoka University. It gives me great pleasure to welcome you to this session of Beyond the Classroom. Ashoka University, as you know, is a liberal arts university funded entirely by private and collective philanthropy. We aspire to make a difference in the field of higher education in India. Beyond the Classroom is an entirely online lecture series. It helps us take the Ashoka mode of pedagogy literally beyond the classroom and share it with the outside world. In a way, this is our method of making our teaching and research accessible to a broader audience. We think that this is particularly relevant in the present times as the world is engulfed in a devastating pandemic, and each one of us are isolated in our own ways, not only physically, but often also psychologically and emotionally. Alongside Beyond the Classroom, there are also several other online ventures the university is in the process of developing. At Ashoka X, a newly developed wing of the university, we are currently devoted to the process of creating and offering full online courses in at least 2 formats for a wider audience. For further details about this, please visit our Ashoka X webpage. Today, we have with us Dr. Imroze Khan. Dr. Khan is an assistant professor of biology at Ashoka University and DBT Wellcome Trust India Alliance Intermediate Fellow. He is an evolutionary biologist and with a special focus on understanding the selective forces that determine adaptive evolution of immune responses. Dr. Institute of Science, Education and Research, Kolkata. Before joining Ashoka, Khan was a postdoctoral fellow at National Center for Biological Sciences, Bangalore, and a visiting researcher at the Free University of Berlin. Today, his lecture is titled Bugs and Epidemics, Darwinian Dilemmas in Emerging Infectious Diseases. Before we begin, there are a couple of things for about half an hour. After that, there will be a discussion. We invite you to engage Dr. Burgers with me in questions using Q&A format in Zoom. That is at the bottom of the Zoom. There's a little thing for Q&A. Please put your questions there and not in the chat. You can choose to share your name or send in your question anonymously. We record all the seminars of Beyond the Classroom. You can find them on our website for those who may have missed them or may want to revisit them at a later date. We will share the link with the entire series with you soon. And now, Dr. Khan, the floor is yours.
Okay, I'm going to start with the obvious question. I hope my slides are visible and I'm audible. Am I audible?
Yes.
Thank you. Sorry. So hello, everyone. Good evening. I want to thank the organizers for inviting me for this talk, and thanks a lot to Just for introducing me for this talk. So for my today's lecture, this is going to be on bugs and pandemics. So I don't want to overload with the information that what this pandemics can do to our lives. I think we all had our own share by now. But today's talk is going to focus more on why and how pandemics take place and what are the natural and ecological contexts in which infectious disease might occur. However, I'm quite happy to, I think happiness is because of the following reason that I don't have to spend a lot of time in introducing my topic, because due to this COVID-19 thing, I think this audience must have already been aware of certain basic terms and terminology that we often use about pandemics, infection, disease. So therefore, it allows me to get into my question directly. So I want to start with this question immediately, and which is the most pressing question of the hour, and which is that how do these new pandemics arrive? Well, I don't mean to give an answer for this because I don't know, honestly. And I think There are many different ways epidemics can arrive, since there are so many different pathogens in this living world. There are so many different animals, and they do interact in many different ways. So pandemics can arrive in many different ways. So, but the idea is to give you some flavor, that to give you some of the natural examples in which some famous infectious diseases have occurred so far. For example, if you think about this Ebola virus outbreak in 2013 in a village called Maibout, which is by the shore of it's a river called Evian, near the northeastern side of the Gabon, which also share the border with the Republic of Congo. So there's something really interesting thing happened there. So it was about around a group of people from the village. They went into the forest to get some food, to get some bushmeat, which is also an important protein source for people living there. So they could not secure any game, but what they found instead is a dead chimp. And it's not a fresh, it's not that the chimp has died recently. It has been a few days. It was rotting already, but yet that's an important source of food. So they brought it back, and that's where that's what people, the future, the further analysis have revealed that maybe that chimp contained the Ebola virus, which infected all these people, and more than half of them, they died. And not only that, that chimp, scientists, they found evidence of around several gorillas also, were found dead. So that is how people start to narrow down, maybe this primates where they died because of the infection, and humans got in touch with this chimps and gorillas as a form of food, and that's how they got infected. So there could be some other examples. If you think about SARS, which happened in 2006, if I'm not wrong. So what happened, it came from some people who were handling the poultry meat, and in a place in China called Guangdong, and from there the disease came to Hong Kong, and from there it actually really became international, because they traveled, an elderly lady, she took a flight to Toronto. So the virus traveled from Hong Kong to Toronto, and within a few weeks later, what happened? Another person, she flew back from Toronto to Philippines. So within a span of few weeks, you can see how this disease traveled almost halfway across the world, and this also tells us the speed at which this disease can spread. But these are some of the recent evidence, so not to say that we haven't had some historical encounters with this infectious disease. So if you think, I think one of the earliest document has been there in ancient Greece, when so people are not exactly sure whether that was plague or not, some plague-like disease. The symptoms were very similar, that happened in Athens, and that actually had a humongous toll on public life. Not only that, it actually killed, it kind of destroyed their army for Athens. And what happened, this also had a consequence in a war that they were fighting in Spartans. So they lost this war, and this is how one of the greatest power of the ancient world actually brought down to its knees. And it has a huge cultural and societal impact, and so much so historians wrote numerous amount of documentaries, so the chronicles, essays, books, novels, and actually people raise this question, that was there ever a disease with a greater cultural impact than this? We all know about the Black Death, which the wrath of this kind of diseases continued even to the medieval era, where almost 1/3 of the population of the European continent, they were wiped out because of this bubonic plague. So we have an example of how plague, shaped up and also influenced enormously the Ottoman Empire expansion, because they had this enormous problem handling this disease. And because of this continuous, handling of the disease, continuous experience with the disease, they also started inventing newer ways to prevent it. So it says that, I think somewhere down there, people actually came up with the modern ways of, the modern ways of public health were kind of took birth in Ottoman Empire. So, for example, the first noted evidence of, how people are quarantined, it actually exists in the document. And people also understood, people actually tracked down that, it's a rodent population, because in many cities, they found that wherever you have abundance of the rodents, the diseases are most likely to occur there. So there's a direct formulations being happening so that's what we are learning about the disease. And, therefore, important question is that also that, what lessons did we learn so far? And as you can imagine, the first thing which comes to our mind is also the species interaction. How different species, they can come together and affect the emergence of a disease. So this is a very famous picture. You all must have been correlating with this. But then you see that a human is interacting closely with rodents, with rats, right? But then in the recent time, this species interaction has got a newer dimension of attention, because due to the rampant urbanization and land use change, because of which many forested lands and ecosystems are being destroyed. And previously, whatever wildlife are living inside the forest, because of this land use change, they're now getting in touch with humans more directly. You see this picture on your right side about how different animals can come into coexistence, where come into the contact with humans. And therefore, the urbanization and land use change has an enormous impact on how how infectious disease can happen. So this is an example. Well, what I just said, it has also been established using certain field studies. The studies have established the direct correlation between the forest fragmentation and the way this Ebola virus disease outbreak happened in Central Africa. Now, if you look at this map very carefully, you see these brown patches. This is where the fragmentation of forest happened. And if you follow, can you see my cursor? So if you follow my cursor here, and if you see this yellow triangle, this is where these are the regions where the Ebola virus outbreak were reported. And you see there's kind of quite a strong correlation between places where fragmentation happened and also disease also took place. So this gives us some sense of ecological information about how disease might happen, and maybe the forest fragmentation and newer contacts between human and wildlife are leading to this emergence of diseases. So I can give you more specific examples in the same context. If you think about the 2013 Ebola virus disease outbreak in Meliandou in Guinea, in Africa. So what happened? Disease started with the disease actually started with the death of a 2-year-old small boy, who was happening to, well, I think people traced back certain incidents, and they say that the child was playing near a tree where there are also a large colony of bats. So, and one assumption is that maybe the child got the disease from bats, because bats are already known reservoir, known carrier of this Ebola virus. And one important fact about bat, which we are also going to talk in detail in sometime later, they also happen not to be reacting towards Ebola virus infection like humans who succumb to the infection. Bats, apparently, they don't have the disease outcome. They don't fall sick. They don't succumb to the infection. And there are also evidence. Well, the way they narrowed down to the bats is the following way, because they were comparing that many species, like for example, they were estimating the carnivores, the chimps, the primates who were there in 2003, and how they were changing the time when the disease came into existence. As I said earlier, that the primates, for example, even different carnivores, they are susceptible to the disease. So if you see that there is no change in the number of animals in the nearby region, so which says that the Ebola virus outbreak was not affecting the other carnivores. Instead, they found this coexistence with the bats who happen to be the carrier of a disease. So this is how people have been looking at the ecological information to find out evidence for how this altered species interaction in the forest, fragmented forest land where you have human settlements are encroaching into the forest, they are causing the disease. Another very interesting example that many people are now thinking as a possible, for example, dogs can also be a potential carrier of the disease, and this comes with a special context. If you think about different forest lands in India, if you think about, let's say, the Jim Corbett National Park in Uttarakhand or Bandipur National Park in Karnataka, you'll find that there's a forest, the land of the national park is a conserved region, and then around the national park you have very dense human settlement, lot of villages around. And these villages, they have a lot of dogs. And what happens, these dogs who also happen to be quite an important predator, they actually in the daytime, they can get into the forest, they can kill wildlife, right? So they can get into touch with the wildlife, not only killing them, but they are getting into contact with the wildlife. They're entering the forest. In the evening, they're coming back to the village and sitting next to you. So therefore, they're forming a link between wildlife and human settlements. But then this kind of potential, this kind of possibilities needs to be experimentally verified. The second important lesson that people are slowly learning, so people were not bothering about this kind of events, but now people are getting more concerned about certain findings which says that pathogens that cause disease in other hosts, it could include humans, it could include some domestic animals, they fail. Well, so in other hosts, they fail to produce any pathology in their natural host. For example, as I said, the Ebola virus, it doesn't kill the bat. It doesn't affect the bat that much. But then when there's the same virus, they enter into the human host, it causes a disease. Now, what is the reason behind such a divergent host pathogen outcome? So, for example, as I just said, the interaction between Ebola and free-tailed bat gives a very distinct example how these bats are not being susceptible to virus. There are other examples. For example, if you consider about hantavirus, which is also sourced from rodents. So they don't cause a disease in rodents. But when this virus enters into humans, it causes a lot of problems in humans, a lot of disease symptoms coming, which relate to the, which cause a disease for their, it affects their lungs and causes disease. And one of the classic example is also the AIDS. If you think about this, the picture is also from, so this is a monkey species named as Sooty mangabe, which is again found in Africa. There are other monkeys also, for example, green monkeys. They happen to be a peaceful reservoir for simian immunodeficiency virus. So now this virus is also an ancestral cousin of the human immunodeficiency virus, which we all know of. So they lead to formation, they lead to AIDS. But then the same virus, which are actually naturally found in these monkeys, they don't cause a disease. So monkeys seems to be invincible. They don't have AIDS, but when it enters to humans, they call AIDS. And I don't have to tell how important this, how impactful this disease is, because by 2013, there are almost 60 lakh infections across the globe. So there are that many ailing humans. So of course, it's a great public health concern. So what we can conclude from this. So one possibility is that since these viruses, these pathogens, they don't cause disease in the natural host, one possibility is that do reservoirs have stronger immunity? So this is a very important question. Or in other words, do humans or other domestic animals, they lack the immunity, they lack the suitable immunity against those pathogens? Is it true? But then when people were doing experiments, they found it's not true. So often disease symptoms, disease phenotypes are not caused because we don't have the immunity. It's actually otherwise. If you see this example of H5N1 influenza virus infection, which is again sourced from birds, right? If you see, the birds are more or less asymptomatic to the infection, and you look at the immune response. So they have low cytokine levels. Again, cytokines, let's not focus in the terms and terminology. It is again an important immune mediator, signaling molecule, which leads to immune responses. But in contrast to what we predicted, what scientists predicted, they found that in the reservoir species, the natural host, they have lower cytokine levels, and they do identify the virus when they enter, but the immune response is not activated. If you think about the intermediate host, sometimes so before they get into the humans, they also get into some intermediate host. If you think about the biology of intermediate host, they cause a mild to severe infection, and the level of cytokine production is somewhat increased. However, in contrast, if you look at humans, it actually causes severe infection. It causes systemic spread, and humans are known to show very high levels of cytokines, chemokines. These are again all molecules which are responsible for immune response. And in the human blood, you can find a lot of acute phase proteins. And this is something we know as a catchphrase called cytokine storm. I think this is a term which is circulating in the media, in the research community, very widely nowadays. Everybody is aware of what is happening in cytokine storm. And it's a response, it's an immune response, which is quite counterintuitive. So practically the newer hosts, the human hosts, are not dying because they don't have immune response. So there's something horribly going wrong in terms of the regulating the immune response. The immune response becomes overreactive or exaggerated, which is perhaps not needed. And it leads to severe consequences. Even in case of COVID-19, we know that apart from affecting the lungs, it can cause severe damage to liver, heart. So therefore, the damage can be caused by the immune response itself. So this again, a little bit more detail. So to just take you through what this image says. So let's say this is the lung epithelial cells, and it is now infected by the virus. You see the term that I was using, cytokine. So you see that lot of secretion of cytokine, and it's in such a huge amount. That's the reason why people call it cytokine storm. And cytokine storm, as I said, is an important immune signaling molecule which signals the immune response. And after the cytokine storm is generated, it also activates enormous amount of immune responses. And they produce a lot of immune molecules which are causing the disease symptoms. And one of the very conclusive evidence came from a study where they were infecting the mice with influenza virus. So if you see the normal mice with normal immune response, they die. Rather, when you see this, so this is a treatment that the group of researchers did. They suppressed immunity. They suppressed immunity somehow, and the result was quite surprising. It's actually the mice with immunosuppressing, the mice where the immune response has been suppressed, they appear to survive more, which again suggests a causal connection between cytokine amplification during influenza and disease outcome, disease phenotype, or the formation of disease. Okay. So therefore, I wanted to create a context by which I can highlight you the difference between the more effective immune response in natural host doesn't mean that it has better immune response or it has higher immune response. But effective means it could be more balanced, because if you think about a cytokine storm, it's created by overreactive immune response. You have immune responses that you don't need. It's an excessive, which is why we call it misregulated immunity or deregulated immunity in new host. And this is what you can differentiate. Based on this information, you can actually differentiate between the host, which are known to be reservoir for the disease, versus a novel host, which is the human host, let's for example, right? So one can hypothesize that maybe when you talk about immunity in a natural host or a reservoir host, so one hypothesis is that they are better at tolerating the pathogen and effects of infection. They can withstand, they're more resilient to the effects of infection. And this is achieved through lesser immune activation, a form of balanced immunity. Again, I just want to reiterate once again, and in novel host, in contrast, what happens? You find overreactive immunity, which can cause severe damage to key organs. Now, this is where the big question comes. What drives such variable infection outcomes? Whatever we have been discussing so far, this is based on observation. But we also need to know, which is where we lack information actually, what drives such variable infection outcome. And this is where possibly the answer lies. One potent hypothesis that people are generating now, it is because of the long-term coevolutionary history of reservoirs or natural hosts and zoonotic pathogen, which is what leading to this very different infection outcome. How it can happen? So, and when I talk about this long-term coevolutionary history, it can be nicely depicted through this interesting cartoon. Again, this is a cartoon from the Red Queen is talking to Alice. This is a character taken by a very famous novel by Lewis Carroll, Through the Looking Glass. And what Red Queen tells Alice is really particularly interesting. She says that now here you see, it takes all the running you can do to keep in the same place. It talks about an inherent interaction between the persisting interaction between reservoir and pathogen that is going on in the natural world. So what happens when a natural host is being infected with a pathogen? They will obviously try to adapt, but pathogen is not going to sit idle, right? It has to also devise a counter-adaptive measures to overcome host response. Otherwise, pathogen will be wiped out. So to overcome that, pathogens also counter-adapt against the natural host. And this creates an arms race. This becomes a continuous process. So what happens if you think about if you come back to this cartoon here, what you see, the Red Queen has a wheel instead of the feet. So she has to continuously move in order to balance herself, right? It's a continuous. So if you think about adaptation and counter-adaptation, it becomes the arms race. Both the side wants to outcompete the other part. So that's the reason why that's what is going on in reservoirs and zoonotic pathogens, possibly. So to give you more details, now we consider this certain animal species which has been, we have, animals which are kind of they have been predicted to be a good carrier of the pathogen. This is what has possibly gone for a long, long time. So they have been going through this relentless process of adaptation and counter-adaptation with the virus for a very, very long time. And we can imagine about a treaty which has been signed over the evolutionary timescale. It is a product of co-evolution. And this is what might lead to very different kind of immune responses in those natural hosts. In contrast to humans who has no known history. So even if you think about the certain coronavirus, which is found in many other natural hosts, so these coronaviruses are not in humans. So humans are not exposed to these viruses for a long time. So therefore, humans or many domestic animals, we don't have required physiological system to withstand or counter the infection. So this is where the critical differences lies. And what you see in bat is also that because of this continuous process of adaptation and counter adaptation, this is leading to very different kind of immune responses. Now people know there has been extraordinary expansion and peculiar diversification of bat immune genes. Not only that, they have also evolved very complex and inhibitory signaling potential in bats, which can control the immune response. And possibly this is what is contributing to the better ability to tolerate the infection. Right? I just want to give you another side to it, how this concept, the hypothesis, can also cater towards our understanding to emerging infections. Now imagine this rodent species here, right? And this rodent is infected with certain pathogens. So this is the outline of the rodent, and then you see this, cartoons which depicts a virus. One possibility is that, like humans, like a novel host, like a new host, they go for very, very high immune response. They just want to eradicate the infection. What happens? They might resist the pathogen by immune activation, and while doing so, it actually clears out the infection. You see that the overall decline in the number of the circulating pathogens. But as we just tried to establish earlier, that how tolerance could be a more probable strategy in this, in this different natural host, because if you think about the tolerance, right? So you see this particular, the second possibility where they're showing pathogen tolerance. What it does, it lengthens, it kind of extends the infectious period. So the circulating viruses or pathogens, they can persist inside the host without being removed. And that gives them enormous, that actually gives them sufficient time to really show very cool genetic changes. So, and that also explains why you might see very high pathogen abundance in rodent species, or maybe some other. So this is an example. This can also be extrapolated to other, other hosts who are natural harbor of different pathogens. Now, if you look at this side, this is something really interesting. So you see this virus here, it's a white one. And now you see in this host, after it started tolerating the infection, it is now has created, it's the same virus, which has now produced different variants. And this can happen through continuous round of mutations, alteration in their DNA. It can go through many different kind of genetic recombinations to produce closely related, but yet different variants. And I'm glad that I don't have to explain this, because you all know that, this virus can, even coronavirus, you can expect many different kind of variants of the same virus. So the whole concept of tolerance allows us to envision a situation where the same virus now can exist in abundance. Not only that, they can also have a lot of genetic variant. Now, this might have a really remarkable impact on how it might become an emerging infection. Now, if you consider a natural scenario, not all the pathogens are able to infect a new host. So if you think about different species, let's say rats and humans, whatever pathogens you find in rats, all of them cannot infect humans. It's those rare pathogen or rare pathogenic variant which causes the infection. And tolerance, since it's kind of, it accommodates both high pathogen abundance and high genetic diversity, you might expect one of those variant might have the ability now to overcome the species barrier. So you now think about the red variant is now have the enough the required genetic makeup, which can be spilled over to a new host. In this case, it's a human, but this kind of spillover can happen between any animals. Even this can happen between different population of the same species. So, but then the whole idea of tolerance and increased pathogen abundance and increased genetic diversity can support the whole logic of how infections can be emerging over an evolutionary time scale. Sorry. And there are, and just I think when I was recently reading about some of the literature I found in October 2020, there is a study which has now established that is indeed an evolutionary arms race that what we just talked about is going on between the virus and host, the bat in this case. This is the broad-nosed horseshoe bat, which drives the genetic diversity in bat severe acute respiratory syndrome related coronavirus spike genes. Now you know that the nexus appears conspicuous, but the large gap exists in our understanding that how to track these links are being formed across evolutionary time scale, because unless we know how these processes are being shaped over an evolutionary time scale, we cannot predict and intervene future emerging infections, right? How do you distinguish between populations which are resistant to infection versus tolerant to infection? Because the population which is resisting infection, they're unlikely to be the reservoir host and be an efficient source of emerging infections. Whereas in contrast, the tolerant host can support many pathogen variant, of which some of them can cross the species barrier and cause the disease. So this is where the field is now trying to move. People are trying to come up with an experimental framework to understand how this evolutionary process is being shaped up. But as you can see, that you cannot go back an evolutionary time scale. You cannot start these interactions in the nature, how bats got the disease for the first time. So therefore, we have to take an alternative way. And that's what our people are taking up. So here, before I end, I also want to highlight another aspect which is going to increase our problem is the climate change and is linked with infectious disease. In many animal and their pathogen association, people have found that the climate change can profoundly impact the infectious disease outcome by reducing the efficiency of the immune system. And as a result of that, disease outcome can be exaggerated. So you see that the flow chart here, so there are multiple aspects of climate change. Could be temperature, which is one of the major features, and this temperature has a direct impact on how host pathogen will interact, as I said, because it affects the immune system profoundly, and this is also going to affect how this transmission being happening. So of course, the climate change with increasing temperature will affect how infectious disease emergence will be shaped in the future. So in the lab, we are also tracking these problems, and we have also found in our own experiments that if you see this graph here, so this is where we are trying to test pathogenicity in fruit flies, which is a very important model system in biology. So in the fruit flies, when they were infected with a pathogen, you see the fruit flies which were living under a warm temperature, they were dying way more than the control, compared to the flies which were reared in a standard condition. So this, if you follow the red lines, their mortality is super high compared to the controls. Similarly, if you look at this graph, the y-axis gives you different treatments. For example, standard condition versus warm condition, and y-axis gives you the pathogen load. You see in the warm condition, the flies which were reared in the warm condition, they have very, very high amount of pathogen load, which again indicates us very clearly that how certain features of climate change are going to impact the immune system. And of course, they're going to affect how they're going to impact the evolution of immune response, and also how they're going to impact the infectious disease outcome. So this is what we're doing at Ashoka to counter some of the problems. So we are investing a lot of effort and energy to understand how the host evolve with and against new infections. Again, tracking back how it started. So as I said, it's incredibly difficult to track in the field because the process has started maybe a million years back. We cannot go back there. So we have to come up with an alternative measure to see or track evolution of host and pathogen interaction in front of our eyes. We have to look at as directly as possible. So we are doing this, we are experimentally tracking evolution as well as genetics of resistance strategy versus tolerance when these interactions are going on. Subsequently, we are also extremely bothered about how does this changing climate and ecology affect such processes. So we are also incorporating certain natural context. For example, how might this entire processes while hosts are evolving and against a new infection being affected by, let's say, climate warming, by increasing temperature, or maybe the lack of nutrition. So as I said, it's going to be a very difficult task to reconstruct the whole process in the natural world, but we are taking an alternative way where we are doing the controlled experiment. So we have animal population in the lab, and now we are subjecting this animal population and with subject under this animal population to chronic infection across generation. We start from the very beginning. We start from the generation 0 when the first time, for example, these animals were infected with a pathogen, and every generation after infection, whoever survives. So let's say if you have 100 individuals, you have an infection. So 60% of them will die, and you have those 40% living. So 40% apparently had the ability to withstand the effect of infection. We are going to create the subsequent generation with the population, with the individual who are remaining. And this process, so they are the one who are going to start the next generation. And this process keeps on going. This continues for multiple generations. And we have the access of such populations which are evolving with infection for more than 150 generations, which gives an enormous ability, that gives a fantastic system to tease apart how this evolutionary process might happen, in natural world also. So this is again headed by a fantastic group of researchers in my lab, which I call Evolutionary Immunology Lab, and they have been doing, many of these people are doing fantastic research to track these problems in the lab to understand the implication in understand evolution of immune response to find implications in infectious disease. So I'm happy to, well, I did not talk about much details, but if you have any questions, I'm happy to take the questions. But if you want to know more about the details, please visit our lab website, or you can write an email to me. So I will end here. Thank you very much.
Thank you so much, Professor Khan, for that amazing talk. I wish I would have seen this 9 months ago. This is just so enlightening. We do have the time for Q&A, so if you have questions, please put them in the Q&A box, and Professor Khan will field them. Just to lead off with a question, this is a total novice question, but how do you imagine doing the experimental setup for identifying the pathogen that could possibly hop to the humans? What would that experiment? I could see how you're doing the experiment in the lab now, but what is the next step in sort of predicting this? I know that that's sort of a far on the horizon, but how do you imagine that setup would work?
That's a fantastic question. So I would say that, first we, okay, shall I stop sharing my slide? Or
yes.
All right. Yeah. So as I said, one of the major challenges is that we still don't know. For example, the whole argument that I had for this tolerance of infection versus resistance to infection, this is something we need to know, that whether this evolutionary process are indeed possible or not. So our experimental paradigm would at least, give us an idea that whether it is a possibility. And not only that, and if it is successful, then it will definitely, will be a very direct, stab at the problem that we are considering. And then it will also give us very important genetic mechanisms which are required to overcome such problems.
Thank you very much. I think a related question has popped up. Are there known markers that make pathogens likely to hop species barriers?
Exactly. I think this is, of course, a very relevant question, and that's what exactly people are doing, trying to track this kind of thing. It is very difficult to, focus on one particular marker, because, the relevance of a pathogen, this is incredibly important to understand. The relevance of a pathogen keeps on changing at the, with the different ecological context. So the same way, so there cannot be a consensus. There cannot be a one marker which would make a pathogen, or pathogen will be able, or pathogen being, enable it to jump the species barrier. But then this is what people are trying to figure out, that whether there are some sort of consensus in it. It's not, but conceptually it looks very difficult. And at least experimentally it is possible to, well, I think in the natural context it could be incredibly difficult to predict these things. But in the experimental setup, we can track this problem. At least this will tell us that whether in nature, whether this happens or not. To pathogen which evolves a novel feature to overcome the species barrier.
Thank you very much. There's two more questions in the bank. I'll do them in reverse order. Prabhav asks if you could explain the difference between resistance and tolerance once again. And I guess piggybacking on that, then, would you know if the Ebola virus in a reservoir host can divide in it? So I guess, building between those two questions, if that would be possible.
So to answer the first question, well, the resistance is the ability of the host to clear the infection. That means you have an infection and you are clearing out the pathogen, you are resisting the pathogen. So you are not allowing the pathogen to live inside the body. In contrast, tolerance is the ability which withstands the pathogen number. So that means you are coexisting with the pathogen. You are allowing to circulate and also withstanding the damage, whatever damage it could do. You have the better ability to withstand the damage caused by the pathogen. I don't know, was it clear?
I think so. And then the related question about the Ebola, if the reservoir host can divide in it, I'm not sure about the phrasing of that.
So you mean that whether Ebola virus can grow or it can replicate inside the host? Well, I'm not getting exactly what it means to be called divide. So does it mean the replication or amplification of the virus?
Yes, I think Rama. I think that's right. Yeah.
Well, I think there are evidence where, of course, I think they can grow inside the reservoir host for sure. But well, this again, I'm talking about from my experience with looking at other pathogens. But exactly whether Ebola can, does it or not, I am not exactly sure. But ideally, they should be able to. So if it is found, because nobody has, well, I don't know, people have looked at whether they can actually divide inside a host, but I think there must be some evidence. So yeah.
Our next question also comes from an anonymous attendee. Does the loss of biodiversity from a patch of forest increase the chance of spillover, as pathogens would be looking for new hosts as their natural hosts keep declining?
Yeah, so that's what the hypothesis is. Well, there are a lot of mixed suggestions. As I said, there has been cases where for example, the graph that I showed initially, that we found, I think the group of people, the scientists, they found that more abundance of Ebola virus outbreak at the places where fragmentation was happening. So, which definitely means that there's a chance of spillover, right? So of course it can happen.
So relatedly, how much do you know about COVID-19? Was that also a spillover event, or could this have been had another source? This is what
spillover is a very generic and broad term. So whenever a pathogen is shifting the host, for instance, where people are guessing that it might have come from bat. So if it is so, then whenever the virus jumps from bat into the human host, definitely it can be regarded as a spillover. Because the virus isolate had very high similarity with the virus circulating in the certain bat species. So definitely can be regarded as a spillover.
Just a thought. I mean, I had read that it was related to the pangolins. Is this not the case? Then it was more likely that it came from bats, the COVID-19.
Sorry, can you repeat it once again?
Yeah, so when this initially hit, part of the argument was that it may have come from pangolins.
Oh yes, there's some confusion.
Yes,
there's some confusion between that was going on. I think they also found the virus, which was isolated later from humans. They also had a lot of similarity between pangolins. So there are also hypotheses that it might have come from pangolin, or pangolin served as intermediate host. So yeah.
The next question is on herd immunity. Is it safe to call the bluff on the herd immunity argument regardless of the current COVID-19 crisis? As you pointed out, the complexity of diversity of pathogens in the natural host and how incomprehensible it would be for the future crisis. So I believe the question there is it better to simply go with a herd immunity argument in terms of dealing with?
Sorry, can you just ask it once again? Your voice broke in between.
I'm sorry. Yeah, the phrasing here, what I gather is, does it make sense to, despite the results we've seen with COVID-19, is it better to simply try to attain herd immunity rather than to isolate a virus, because it's just going to be more and more diverse. It's going to find new ways to spill over. I think that's the question.
Still not getting the question. Maybe I'll just read. Is it in a chat box?
Yeah, it's in the Q&A. It's by CL Kabilan.
Okay, as you pointed out, the complexity of diversity of pathogens in the natural host and how incomprehensible it would be for, I think these two things can be separated out. So herd immunity has been a concept which has been there in the field for some time, that, you allow the population to gain some sort of immunity against the given pathogen. So this is possibly, it's again a very important societal question, but in a way, this is not difficult to find in the natural host and pathogen system. So of course, it does happen, but but I can't say whether it's actually can be implemented in humans or not. So that's a debatable issue. But yeah, but it's definitely a relevant biological possibility.
Speaking of diseases more generally, has there been an example of a disease for which humans have developed tolerance and resistance over many generations that initially it wiped humans out, or at least was more deadly?
Okay, I'm sorry again. Can you just repeat the question again?
I'm so sorry. Yeah, so there's a question from anonymous attendee which reads, historically, has there ever been a disease where humans have developed natural resistance / tolerance over generations after succumbing to it initially?
Well, I think there are some historical evidence. For example, if you think about the Europeans who traveled from Europe to the New World, so when they went as a conqueror to the America, so they took the smallpox with them. So apparently they were known to be tolerant to the disease because they had a disease for a long time. It was existing in Africa and Europe for a long time, right? So you could assume that they were tolerant to the disease. But then when they went to America, when they were exposed to Indians there, so they had not seen the virus ever in their life. So it actually created havoc. So this can be interpreted as one such case where one human population were tolerant to the disease, and when they interacted with another who did not have the immunity against the disease, they suffered. So yeah, so I think maybe that could be one such example that I can think of right now.
And Prabhav asks, if a species is tolerant, would the pathogen be harming that species without the species exhibiting symptoms, or would the pathogen not affect the species at all?
Again, so yeah, I think I also defined the whole thing in a more simplified version, but then I think if you want to think more deeply, it would be rather more like where they are better at withstanding the effect of infection. So there might be some effect, of course there might be some effect, but even if you think about, so let's say just forget about a host which is tolerant, which is tolerant, or which could be tolerant or resistant, so whatever be the case, so let's forget about that. They have evolved to be tolerant or resistant. But even if you think of a population level, So different population have different response to the pathogen. Some population might be more tolerant, some population could be more resistant. So the comparison is at the level of more and lesser. So it's always going to be relative term, right? But then when you're tracking a given host and pathogen, you have to experimentally show whether it is actually true or not. So what I'm trying to say, what I tried to say in the talk is going to an assumption, and maybe the disease symptom in those natural host is milder because they're better at tolerating the infection. So, but then they can definitely have some negative consequences, of course. But again, it's a relative term.
And I think we have time for one more question. You already covered this in the talk, but perhaps maybe you can unpack it just a little bit more. Is there any effect of climate change?
So you mean that is there any effect of climate change on infectious disease? Oh, yes, I think that's what I was trying to summarize. I'm sorry that if I went a little faster there, but yes, the climate change, the certain component of climate change also, for example, if you think about climate warming, which is again an important feature of climate warming. So global rise in temperature. And temperature is known to affect immunity in various ways. One significant way by which temperature can affect immunity is to reduce the efficiency. As you said, an example that, the population of the fruit flies were living in the warm temperature, they were dying more compared to the ones that were reared or the one that were, lived in a standard condition, or, standard means, the normal condition. So their mortality was higher compared to the standard condition flies. And they also had higher bacterial or higher pathogen load. So of course, it suggests that the immune response, or immune system has gone or has been compromised. And this will of course affect how, and let's say if your immune system has gone poor, and if you're again encountering a new infection, right? So if your immune system is not efficient, then you will definitely suffer. And this is going to affect the population as a whole. And this will also have a long-term evolutionary changes, because then the organism has to evolve newer strategy, newer way to start countering the pathogen when the temperature was high, because its immune response has gone bad. And the question is whether they can do it or not. There are some depressing results that people are talking about. Let's say if you compare the birds in Europe versus birds in the same population of birds in Europe versus same population of bird in Africa, it seems that if you put the European birds in the African climate, the temperature is very, very high. So they simply fail to evolve an immune response. So that's not a great news, but we have to do more experiments to substantiate this claim.
Thank you so much, Dr. for the lucidity with which you answered all those questions. I do want to note to all the participants that we will be doing a quick poll at the end of this, so please stick around with that. We'd love your input on these seminars. So as we come to the close of today's seminar, a word on our upcoming offerings. There are a host of 6 live online courses on offer this December and January on what we call the Ashoka Academy. The registration process is still open, and you can register on Ashoka on our Ashoka X webpage. We would absolutely love to know what you thought of today's seminar, so please do fill in that poll. On behalf of Ashoka University, I would like to thank Dr. Imroze Khan for this amazing presentation. I also extend my heartfelt thanks to all the members of the audience who joined us this evening, and also for all your wonderful questions. Thank you very much, and enjoy the rest of your day.